Good morning. Welcome back. Great. Exam 2 results are posted. This is the raw scores. Okay, so I don't want you to get super discouraged. Like the average is like 55%. I know like everybody's coming out of high school. Everybody is like top in their class, right? Getting 90%, 95% on all the exams. That's not the way it works usually in courses like this. So you don't have to worry about it if you didn't get 90%. You don't have to get 90% to get an A. Actually, you'll know you need like 70%. Okay, so it's okay. And you know, we'll see the average will vary from test to test, right? And just it sort of just depends on the overall difficulty of the question and how people did. And all of these exams are normalized across the different sections. So if one section has an easier test than the other ones, that doesn't give you that section an advantage over anyone else. Okay, that's just how it works. So don't worry about it. You can take a look at this. My exams will probably be pretty similar in difficulty to Professor B's. Maybe easier. Usually my averages are higher, let's see, but not that much higher. So I will try. I'll do my best. Okay, so what we're doing today is we are going to get back into the actual content. And this is going to be, I think, in many ways, the immune system is the second most interesting thing, I think, about the body. Because it is so complex, so rich, and we don't fully understand how it works yet. And it's so incredibly impactful on health, right? Like you're basically living in a sort of an adverse environment here, right? You have an incredible array of different resources inside your body that pathogens or the creatures would love to gather those resources, right? And use them to multiply and to, you know, to sort of proliferate their own genetic material, right? It's almost like an arms race. And the immune system is an ability for you to fight off those pathogens, those bacteria or viruses or different things that could potentially cause you harm, okay? Now, for this overall lecture, or the next 2 lectures, we're going to be talking about mechanisms of innate immunity, okay? So these are the tools that you have that are sort of built into your body that can rapidly attack pathogens, things that are trying to give you harm. Then we have aspects of adaptive immunity, which includes things like the generation of antibody diversity. Everybody's heard of an antibody before? Right. I mean, we all went through COVID. We've probably heard of antibodies now, right? So the generation of antibody diversity is really an incredibly sort of interesting thing, right? How can we generate some tool within us that can essentially recognize any pathogen and cause a directed and very powerful response specifically against that pathogen and not against anything else within our body, right? So we're going to have, that is going to then be sort of turned into a variety, those antibodies are then sort of changed into different structures that can be targeted to different parts of your body in order to most effectively attack different antigens, then also we have to understand how, if we're able to generate this diverse ability to attack any specialized sort of structure, how come we don't attack ourselves, right? And of course, autoimmune diseases, things like lupus or something, are sort of situations where our body then starts to recognize itself as a pathogen and attacks itself, right? So how do we avoid that? Okay, and that gives rise to origin of immunological memory, which is the ability to not only can we sort of coordinate a very highly effective attack against a specific pathogen, our bodies remember exactly the shape of the epitopes on that antigen and can attack it again with an overwhelming response, even if we basically are just exposed to it years later. And this is really the reason why things like vaccines work. Vaccines are simply our ability to train the immune system to recognize a pathogen, okay, or a component of a pathogen, and train the immunological memory to then recognize it without you having to actually get sick from it, which is great, okay? And then we're going to talk about blood type. If we have time, we'll talk about a variety of different diseases, viruses like HIV and COVID-19, and how those intersect with the immune system, and different tools that we can use our knowledge of the immune system to develop things like, testing for various diseases or immune therapies that can fight cancer. Okay, so that's a lot of topics for 2 lectures. So I'm not sure how deep we'll get into these, but feel free to stop and ask questions at any time. So first of all, what is a pathogen to begin with? Okay, so a pathogen is really any foreign invader that can cause an infection or a disease once they enter the body. Okay, so these come in a variety of different classes, and we can sort of sort them based on, I don't know, like size, really, okay? Or into cellular and acellular parts. Okay, so cellular are going to be like living things, okay? We've got parasites, like a tapeworm or something that can get in there, can be quite long, right? We have to find some way to fight those off. It's often going to be done by eosinophils. Then we have diseases that are sort of single cell based, but still pretty big cells like protozoas. Malaria is caused by this. We could get fungi that infect us. You could have fungi in your foot, right? If you're, you can get athlete's foot or jock itch, right? Or fungi in your lungs, which can cause a pneumonia. Then we have smaller things like bacteria that can cause leprosy, or can cause, you get your bad E. coli, can cause food poisoning, things like that. And then we move into these sort of categories which are non-living. Virus is sort of like a quasi-living creature. It can't replicate by itself, but it does have the genetic material needed to create a replication of itself. It just has to attach to a living host, which would be the cells in your body. Okay, so HIV is an example of a virus. COVID is an example of, SARS-CoV-2, I guess the appropriate way to call it. And then finally, there's even some pathogens that are definitely not living at all, that are just weird misfolded proteins, which can induce other protein, the same protein to misfold and cause an aggregation. Okay, those are things like prions in Creutzfeldt-Jacobs disease, also known as mad cow disease, which was a big thing 20 years ago. Okay, so these are our sort of our classes of pathogens. What can we do to overcome these pathogens? What are the tools that we have at our disposal to fight these off? And you can classify them into large, sort of 2 major groups. We've got the innate immunity, okay, and we have adaptive immunity. Okay, so innate immunity, all animals have some sort of innate ability to recognize specific traits of pathogens that are broadly shared by broad categories of the pathogens, okay? And these are, the responses here are going to be characterized by a rapid response. We can immediately, there's some common themes that many pathogens have, right? That if we see that, if our immune system sees that, it can immediately attack it and find, it already knows its weakness. And this is baked into our genetic code. We've been learning for, eons about what makes a pathogen like dangerous to us, okay? And we can sort of attack it immediately. Now, there are sort of 2 classes, and then we have this adaptive immunity area, okay? And the adaptive immunity area really takes a few days to turn on, okay? And these are traits that are highly specific to the precise pathogen that is the precise version of the pathogen that has infected you, right? Like the sort of shape of a virus might change like the coat protein around the virus, which makes it unable for your innate immunity unable to recognize. But your adaptive immunity over time can recognize that this particular virus is infecting my cells, causing them to sort of lyse or to making me sick. And your body can train itself to then attack that specific shape of the virus, okay? So that takes time. It says in the book of vertebrates only, but that's not actually true. Even bacteria have some forms of adaptive immunity. The CRISPR system, if you've ever heard of that, that's used for genetic engineering, is actually a very interesting way that bacteria can cause adaptive immune responses. Now, within our innate immunity, we can have things like barrier defenses. Okay, so the first defense against any infection is our skin, right? We have these epithelial cells. They're super tightly packed, okay? They don't pass water between them, okay? And on the surface of our skin, we've got hairs that are going to sort of keep microbes away from our body. We've got secretions, sebaceous secretions here that are oily, that maybe change the pH to a level of pH that is different from what's inside our body, right? If there's some creature that wants to replicate inside our body, the environment inside our body should be very, if we want to protect against that, then we should create a barrier that has a very different environment, so that something that's adapted to live inside us will not be adapted to live outside us, right? So we want to have a different pH, have an oily environment rather than a salty environment. Also, we can secrete things like lysozyme, which can break down cell walls on the surface of our skin, or microbes on the surface of our skin. And then we also have an entire sort of microbiome of little things, bacteria and stuff living on our skin, living in our gut, right? That is well adapted to that environment and provides positive sort of tools for our body to live and outcompete many other microbes that might get onto our skin. Okay, now, so then we also have mucous membranes. Those are going to be at any place where we're interfacing with the external world, right? So we have urinary tract, we have the nose, mouth, throat, the lungs. Those are places where we're going to be secreting a thick mucus that is full of antibodies and that can sort of physically trap these pathogens. And then embedded within many of these mucus systems are cilia, which are going to continue to sort of push that mucus out towards the external parts of our body, right? There's like within our lungs, there's a mucus elevator that sort of is constantly sweeping and moving mucus up so that we can eventually sort of spit out anything that gets trapped in there, okay? Or swallow it into the acidic environment of our stomach, right? Inside our stomach, we have a highly acidic, like pH 1.5, you know, where it's like a million times more acidic than our blood, okay? And so many pathogens are not going to be able to function. Their proteins will fold in an inappropriate way within our gut, or they'll get dissolved completely within our gut. Right, so on the other hand, now, okay, then we have these also internal defenses. So these are barrier defenses. These are going to keep the pathogen from getting in. Then we have internal defenses, which can be sort of categorized into cellular and acellular components. So cellular things are like phagocytic cells. Phagocytic cells are cells that are going to swallow, eat, and then decompose pathogens. Okay. Then we have natural killer cells. They're called natural killers because they don't have to undergo an adaptive immune response to get activated by an adaptive immune response. They can just immediately see a common motif on a pathogen and attack it with an appropriate sort of destroying tool. And we'll look into this a little bit deeper in a bit. Okay, those tools that they can use are things like antimicrobial peptides that might punch holes into cell membranes, or things like granzymes that can trigger apoptosis, program cell death in a pathogen. Okay, and then we also have an inflammatory response, right? You poke yourself, you get an infection. What happens? You get an itch. You get the area starts to swell, start to feel pain there. It starts to feel warm, right? There's a host of different things that are going to coordinate a locally specific response to that infection. It's called inflammation. Then finally, here in our adaptive immune response, we've got a humoral response, so named after the four humors of sort of classic, sort of pre-medicine times, I'd say, right? Where it's basically any of the body fluids, okay? And then we have a cell-mediated response, which are going to be these cells which recognize and are trained to recognize specific, very specific attributes of a pathogen and attack it directly. Okay, so that's kind of a broad overview. So in terms of phagocytosis, right? Phagocytosis, this is the process of, this is a very common way that our innate immune system and our adaptive immune system can end up killing cells. This is a kind of effect, or killing pathogens. It's a very common mechanism. Okay, and the way it basically works is you're going to have some sort of phagocytic cell. A phagocytic cell is just a cell that eats other cells or eats other pathogens. Okay, so it can sort of extend some filopodia here or pseudopodia around here. They're going to get engulfed. They're going to endocytose. And depending on what's inside this vacuole here, or also known as an endosome, depending on what it sees in there, it can send things into different pathways. It can sort what it has sort of swallowed and either redisplay it up on the surface or break it down by merging it with what's known as a lysosome. So a lysosome is a little packet or a vesicle that has low pH, and it has lysozyme and other enzymes that are sort of tuned to destruction of other proteinaceous material. Okay. These are generally kept in tight packets and are not allowed, that material is not allowed to sort of wander all over the cytosol. It would cause total havoc, right? These are things that are going to break down proteins. Okay, so we have, instead we've compartmentalized these things and then allow the lysosome to fuse here with the vacuolar endosome in order to break down specifically what was swallowed, nothing else. Okay, those are then going to get broken down into fragments of proteins and fats, and then that debris will either be will both be released, or depending on the kind of cell that this is, it may even, those fragments may even get displayed on the surface of this cell on a special pedestal that will say, this is what I just ate, and try to coordinate the adaptive immune response to then target those types of fragments. Okay. So how do we recognize all these sort of different, all the myriad possibilities of antigens on the surface of pathogens? How do we recognize pathogens? Well, in the innate immune system, that is done through things called toll-like receptors. Okay, so what's a toll-like receptor? It's not a toll where you're paying a toll on the highway. It's das ist toll, okay, which was what they said when they first figured it out in German, which is, this is really cool. Okay, so this is a suite. There's a suite of toll-like receptors that can live at different locations on your cells. Okay, they could be expressed on the extracellular surface of the cell, like toll-like receptor 4 or toll-like receptor 5, or they can be expressed within vesicles, like those vesicles that we just saw that were in phagocytic cells. Okay, like toll-like receptor 9 or toll-like receptor 3. Okay, and each one of these toll-like receptors recognizes one of those common motifs, structural motifs that are on or part of a pathogen. Okay, so for example, toll-like receptor 4 here, okay, it recognizes lipopolysaccharides. What's that? A lipopolysaccharide, that is a piece of fat that has a chain of sugars attached to it, okay? So those specific chains of sugars on fats are found on the surface of many bacteria. Okay, so if you see a lipopolysaccharide of a particular class, that's not something that your body will ever make. It's the stuff that bacteria will make. So if you see it, if this cell runs into it, phagocytic cell runs into it, it might trigger a response. It will grab onto that, and that will trigger the process of endocytosis and phagocytosis of whatever it just. Another thing that you could see is, so toll-like receptor 5 recognizes flagellin. Okay, flagella are these little, I don't know, they're like little floppy tails on some bacteria or on protozoa that spin around, okay, and allow bacteria to move around the world or move around your body. Okay, so they're also not found in your own body's tissue. So if you see it, you know it's part of a bacteria. We're going to want to phagocytose and destroy that. Okay. So after that phagocytosis process happens, there are other toll-like receptors that are expressed within those vesicles, okay, in the endosomes, that can recognize other common motifs. Okay, so another one here is toll-like receptor 9. Okay, toll-like receptor 9 recognizes CpG DNA. Does anybody know what a CpG is? Cytosine phosphate guanidine. Okay, so we have in our DNA, we have 4 different residues that we can use: A, T, C, G, right? So the C and the G here just refer to a C and a G that are next to one another on a piece of DNA. Okay, and this particular CpG is unmethylated. Okay, so in our bodies, most of the sites that have a C and a G that are next to each other have a methyl group attached to it. That blocks basically any genes that might get turned. It blocks that ability of that gene to get turned on. Okay, and you have to remove. If you remove that methyl group, then that gene can get turned on. Okay, in bacteria they don't really have that. Okay, they don't have that methylation. So if you see unmethylated CpG DNA, that's a very good sign that it's coming from a bacteria and not yourself. Okay, so that's a target for destruction. Okay, and then we have toll-like receptor 3. Another example here. This is looking for double-stranded RNA. Okay, RNA is something that's very important within our own body. We use RNA, right? We have DNA gets transcribed into RNA, messenger RNA, and then that messenger RNA gets translated into proteins. This is the standard way we produce all proteins in our body. Okay, but guess what? That messenger RNA that we make is only a single strand. It's not paired up. Okay, so if you see a double-stranded RNA, that is actually the way that most viruses store their genetic material. Because viruses don't have like a whole cell to work with, right? And they want to be as close as possible to making the protein, they generally store their genetic material as double-stranded RNA instead of DNA. It skips a step in that protein production process, makes it easier for it to sort of produce protein within cells. Okay, RNA is less stable, could lead to more variation, but that's not a bad thing for a virus. A virus is constantly trying to mutate itself in order to invade your body. Whereas us, we will use double-stranded DNA because we want something much more stable. Okay, so these are sort of 4 examples, and here are a list of other ones, right? That basically the whole point here, though, is that specific motifs can be recognized by specific toll-like receptors, and all of them are going to drive the activation of these toll-like receptors. Then it's going to drive a downstream cascade that's going to cause an innate immune response. Okay, now there's some convergence in that cascade, and there's some specificity within that cascade. And I'm not going to put on a slide of that sort of map because it's super complicated and you'll spend a lot of time memorizing it. And it's just sort of beyond the scope of this course. But an important point to make within that is that innate defenses here will have some specificity to them. To your question? Sure.
So on that graph, TLR9, TLR3 are listed on memory.
Great. So this is a great question. I love this. Okay, so TLR9 and TLR3 are only in this vesicle, right? Whereas these other ones are out there on the cell surface, right? Why they target that the toll-like receptors are expressed in specific compartments where that structural motif of the pathogen has the best chance of being revealed, right? So if you are looking for something on the outside surface of a pathogen, you should put the receptor that will recognize it on the outside surface of the cell, right? If you're looking for the double-stranded RNA, that's not stuck out on the outside of a cell, or that's not stuck on the outside of a pathogen. That's within the capsule of the virus. So you want to get to a place within this vesicle here where you're breaking down the pathogen and you can recognize, oh, look what was just inside this little thing that I found. Uh-oh, double-stranded RNA. Let's activate a specific response against a viral pathogen. Yes. Right. The location helps you determine what its function is. There is definitely a link between the sort of functional class of toll-like receptor and what it recognizes and where it is targeted to within the phagocytic cell. So let's look a little bit more about the specificity of these innate defenses, which is just an example here of two different experiments. What's all one part of one big experiment, okay? Where they wanted to see, okay, if you trigger an infection, is the response to that infection specific to that class of pathogen, or does it just sort of turn on a general defense? Okay, and this is how they did this experiment. They did it with fruit flies, Drosophila, okay, and they looked to see how long will Drosophila survive following infection of a fungus, okay? And they have this wild type Drosophila. They infect it here at 0 hours post-infection. And on this red line, how many Drosophila are left after 1 day, 2 day, 3 day, 4 day, 5 day? And you can see, I mean, fruit flies live like 3 weeks anyway. So some of them are going to die anyway, but they're pretty resistant to this infection, right? They are not dying off, okay? Now, if you have a mutant fruit fly that is mutated so that drosomycin is the gene drosomycin cannot turn on, it's been knocked out, okay, then this particular fruit fly dies off within this class of these mutants will die off within 2 days following the infection. So they are not able to mount an effective response against fungi, okay? If you try to rescue that, that mutation by giving the fruit fly drosomycin within its like feed, within the stuff that it eats, guess what? Now the mutant, even though it can't turn on the drosomycin gene, since there's drosomycin around, it survives. However, if you give it defensin instead, which is a different class of microbial, antimicrobial peptide, it dies at almost the same rate as the mutant itself. Okay, so what does this show us? Okay, this is showing us that there are specific peptides, okay, specific antimicrobial sort of proteins or peptides that only work on fungi and aren't effective against other stuff, okay? And that these are going to probably be encoded within the genome and get activated specifically by infection, okay? But we haven't shown that part yet. Now, on the other hand, we want to show that this works in both directions, okay? That it's not just specific to drosomycin, might be specific to fungus infections. Is it effective against a bacterial infection? No. Okay. So in this case, now they infect the same sort of set of flies, or they infect the flies now with a bacteria. Okay. Wild type doesn't die because they have an innate immune response against bacteria. Okay. If you have a mutant that knocks out defensin, okay, these will die within 3 days. If you provide defensin in the chow, they survive. If you provide drosomycin in the chow, it doesn't save the fruit flies. Drosomycin only works against the fungi, whereas defensin only works against the bacteria. So both of these things are activated by distinct toll-like receptors and mount a specific innate immune response that the class of pathogen would be sensitive to. So fungi are sensitive to drosomycin, bacteria are sensitive to defensin. But how can we now actually show that the fruit flies actually upregulate the expression of the gene, that they start to produce drosomycin or they start to produce defensin through that sort of cascade that follows an infection? How could we actually show that? Okay, so one tool that is very commonly used in molecular biology and in the immune system in neuroscience is a tool called the green fluorescent protein. Okay, so this was sort of invented by this guy Roger Chen, won the Nobel Prize for it in 2008, along with 2 other people. And this is a tool which is derived from jellyfish. Okay, so jellyfish, they glow, right? Some jellyfish glow. Okay, this is Aquiora victoria. It luminesces and also fluoresces. There was a man, Osamu Shimomura, that shared the Nobel Prize with this, and he would squeeze the jellyfish and try to figure out what's making it glow. He figured out what made it glow, isolated the protein, then a gene was sequenced for the green fluorescent protein. which then was mutated to create different colors of this fluorescent protein, which you can see here in different bacteria sort of smeared on a plate that have different colors of this fluorescent protein. Now, what's cool about this fluorescent protein is that it is a single gene, okay, that we can use and we can put into the genome of another creature, okay, and use it as a tag, a fluorescent tag that then you can see if that gene product is being produced or not. Okay. So this is the experiment that Lamartine did in France that re-engineered the fruit fly genome to demonstrate that an infection will specifically turn on an innate immune response gene. Okay. So in this case, they infected a fruit fly, and first they engineered a fruit fly to take that defensin gene, okay, and replace it with the green fluorescent protein. Okay. So in the sort of the DNA of this fruit fly, this is where the defensin gene normally sits. They cut it out, they put the gene for GFP in, green fluorescent protein, but they put it still right behind this, what's known as a promoter. This is the section of the genome that doesn't make a protein, but controls, regulates whether or not this gene will get turned on. Okay, so whatever sort of cascade happens when you infect an animal with the bacteria, it's going to activate some factor, transcription factor, that will bind here and say, turn on this defensin gene. But guess what? We swapped out the defensin gene with the green fluorescent protein. And now, if we don't poke the animal with a bacteria, if we don't infect it, the animal just looks like that. And when it's underneath blue light, it doesn't sort of shine at all. If we do poke the bacteria, wait a few hours, all of a sudden the fruit fly starts glowing green. Okay. And that is because we specifically, this demonstrates that an infection will specifically turn on the defensin gene. Okay. And we can visualize that now by replacing the defensin gene with this GFP report. Is that clear? Any questions on this? Okay, cool. So, all right. So let's look now at the cellular components of the immune system. Okay, so all of these sort of immune cells here sort of originate within a progenitor cell, the stem cell, a multipotent stem cell that can turn and differentiate into many different other progenitor cells, and then more highly specialized cells in this sort of distribution here. So we have a stem cell. It is going to be able to turn into myeloid or lymphoid stem cells. And from there, the lymphoid stem cells will turn into lymphocytes. Myeloid progenitors will turn into granulocytes. Okay. Granulocytes are so named because they have little granules, like little pieces of looks like sand if you stain them. And those little granules are things that they can release. They're like the lysosome, like lysozymes. They're little packets of material that they are that can either attract other cells, like cytokines, or destroy cells if they phagocytose. Okay, so let's sort of work our way through here. So the myeloid progenitor cells, these granulocytes, are going to be part of the innate immune response. Okay, they have neutrophils, which are small phagocytic cells that will go and swallow viruses or maybe a single bacteria. Okay, they have eosinophils, which can sort of eject granules of enzymes that destroy and target things like tapeworms, these parasites that can live inside you. Basophils and mast cells will release things that trigger like allergy responses. And then finally, we have these monocytes, which turn into dendritic cells and macrophages, which are sort of professional antigen-presenting cells. So macrophages are big cells that can eat many bacteria, whole collection of them. Okay, and dendritic cells are cells that will phagocytose, eat the bacteria, and then display its fragments on its surface to coordinate coordinate an immune and adaptive response. So a macrophage is actually an example of a macrophage. Is this right here? Here's a macrophage that's stained that is sort of phagocytosing a set of bacteria right here. Okay. So those are the earlier early recruitments, okay? Then we have these lymphoid stem cells that these are going to be sort of recruited late into the specific and adaptive immune response, okay? Now we've got lymphocytes, they're going to turn into B cells and T cells. B cells and T cells are so named based on where they mature. B cells will mature in the bone marrow, T cells in the thymus, okay? All of them are going to be born, all of these are born in the bone marrow, but where they mature is different here. And then we have natural killer cells. These can attack quickly. The B cells and T cells are going to be part of the adaptive immune response. And they can be broken down into these other sort of pieces here that we'll talk, or they can differentiate into these other more specialized forms of adaptive immune cells that we'll go into later. Okay. Here's a funny joke. Now, where does this all happen? Okay, this is all part of the immune system. What is the immune system? Okay, we have the primary organs of the immune system are the thymus and the bone marrow. Okay, so inside all of the long bones in your body, okay, like bones are made of calcium mostly, and then they've got hollow spaces within them. Okay, inside there is the bone marrow. If anybody's ever had ossobuco, that's what you're eating. Okay, and that is where all of these sort of progenitor cells will continue to regenerate themselves. Okay, and then in the secondary, what's shown here is sort of the secondary immune organs that include things like the spleen, okay, right here. This is an area where your blood gets filtered and red blood cells or erythrocytes that are damaged get broken down and recycled. The hemoglobin gets recycled. And that's also a site where you'll be screening the blood for pathogens. And then we have these lymph nodes here that are scattered all over the body. And you could maybe consider the tonsils and the Peyer's patches similar to lymph nodes. Okay, and these are areas of convergence of the lymphatic system. Okay, so remember, in your circulatory system, you've got a bunch of blood that's circulating around, and fluid slowly leaks out of the capillary networks. Okay, a lot of it comes back in, but not all of it. Okay, about 5 liters per day sort of seeps out and doesn't get returned. That has to get returned by these other passages of tubes, the lymphatic system. Okay, so you have drainage. There's no like the fluid moves in the lymphatic system back towards the thymus, and it passes through these little nodules. Okay, these are sites where you have a large number of T cells that are going to scan anything that comes through there and for potential pathogens, and can initiate an adaptive immune response against those pathogens. Okay, so you can sort of see a diagram here of the lymph node, right? We have some lymph comes in, and we have sort of it's going to pass across all of the surfaces of all of these sort of these immune cells before the fluid continues on its way. Okay, so if you have a cancer that is growing inside your body, the staging of the cancer is going to be dependent on how far the cancer invaded the blood or it's invaded the lymphatic system. And so maybe if you have a stomach cancer, right? If someone has a stomach cancer, they cut out a piece of the stomach, they'll also sample and biopsy many of the lymph nodes around the stomach to see if there are any cancerous cells there. If you have a cancerous cell that has grown into the lymph nodes, instead of a stage 1 or stage 2, you're now in stage 3 or stage 4. Okay, that indicates the cancer has metastasized and grown to be able to live outside of the specific environment of the stomach. Okay. Okay, so when we get infected, what actually happens? Okay, we have sort of a series of steps that is going to give rise to this inflammatory response. So we've got some sort of pain and swelling that is going to be induced by molecules that are released upon injury or infection. Okay, so we have a splinter that comes in, for example. There's pathogens here. There's toll-like receptors, these macrophages and mast cells that recognize, okay, some motif on here, and then they're going to release signaling molecules that are going to do a number of things, right? One, they're going to cause a dilation of the capillary networks there. Okay, so that's going to cause more blood to flow into that local area. That's why you're going to see the redness and swelling. Okay, we have a locally higher sort of concentration of blood flow there. Okay? It's also going to cause the spaces in the endothelial lining of those capillaries to open up. Okay, that will allow passage of other cellular components of the immune system to migrate into the actual tissue out of the blood. Okay, and sort of attack the pathogens. We can actually image this. This is how this is within some vasculature of the blood. Okay, we have red blood cells traveling very quickly through the center of the channel here, the lumen of the vessel, and then along the sides here, there's these white blood cells, leukocytes, that are slowly rolling along the wall of here. Okay, they are seeking signaling molecules, which will cause them to stop at a particular location and start to infiltrate into the surrounding tissue. So we have infiltration here of these leukocytes, in this case, neutrophils in this example, okay? They are going to get recruited to the site and begin to phagocytose the pathogen, okay? Now, how does, and this is going to then cause the release of antimicrobial peptides, the accumulation of pus, which is like a fluid that's rich in white blood cells and dead pathogens, and also cell debris from the phagocytosis and lysis of the pathogens. And here's an example, and these are all recruited through what's known as cytokines. So these signaling molecules are cytokines, and they attract cells that have receptors that respond specifically to that class of cytokine. So a cytokine is something that can attract other cells, and the cells will move towards this. In this case, a white blood cell is going to migrate towards this pipette that's releasing a cytokine. And this cell will try to like extend the filapodia and actin filaments in this direction, retract them on the other side, and start walking towards that cytokine. Okay. This is specific. There's different cytokines that will attract different classes of cells. Okay. So in an immune response, right, we're going to release histamine and these cytokines that are going to cause neutrophils to migrate into the area. Okay. Then they're going to phagocytose these pathogens, destroy them, excrete the material into the surrounding area, which will then get eventually cleaned up and diffuse away, and lead to healing. Okay. Any questions? Yeah. What's the function of what? Histamine is a molecule that causes the swelling and dilation of the blood vessels. Okay, good question. Okay, that is often released by mast cells, and if you see an allergy, an allergic response is you usually get an overall swelling of your tissues, right? You get that's a histamine-based response that's caused by the release of histamine from mast cells. That's why if you have an allergic reaction, right, or you're bothered by allergy, what drug do you take? Take antihistamine, right? Okay, that is going to block histamine receptors to try to block that process. Okay, so now we're going to transition from innate immunity to adaptive immunity. Okay. So the adaptive immune system has this sort of the core feature of the adaptive immune system is what's known as an antigen receptor. Okay. And we have two different cellular components here. We have a mature B cell and a mature T cell. Okay. And both of them are displaying antigen receptors. So the adaptive immune response is going to rely on these two types of lymphocytes. Okay. These are white blood cells. Remember, the B cells are maturing in the bone marrow. The T cells are maturing in the thymus. Okay. And they both express antigen receptors that have a little bit of a different sort of overall shape to them, but they also have a very highly specific shape to the tips here. Okay. And these are shapes that can recognize a huge variety of potential antigens. So what is an antigen? Okay, that's a special word, right? It's a substance that can elicit a B cell or a T cell response. Okay, so anything that can cause these cells to get activated is called an antigen. Okay? And there's going to be a small part, the actual physical part of the antigen that the antigen receptor binds to is going to be called an epitope. It's usually only a few amino acids in size, okay? And each individual B cell and T cell is going to be unique, okay? It's going to have its own unique shape to the tips of these antigen receptors. And we're going to have a tremendous diversity of these B and T cells so that we can recognize millions of different shapes, millions of different epitopes from each unique B and T cell. Okay, so each B cell or T cell has a different type of antigen receptor binding pocket here. Okay, but all of the antigen receptors within a particular B cell or T cell are identical. Okay, so there's not a diversity within the B cell itself, within an individual B cell. There's only diversity across different B cells. So, and that is going to allow when a particular B cell or T cell binds to an antigen powerfully and gets activated, you're going to be able to amplify and create copies of that specific B cell or that specific T cell to create a huge exponential response against only the antigen that this particular cell recognizes. And this is where the origin of the specificity of the adaptive immune system comes from. So how do we make those binding regions? Okay, what does this sort of antigen receptor look like? Okay, and we'll start here with the B cell receptors as our model for how antigen receptors are shaped. So each B cell receptor, so here we have the cytoplasm of a B cell, okay, and then displayed on the cellular surface, okay, we have our antigen receptor. And there'll be millions of these displayed on the cell surface probably. Okay, so first we have this C region, okay, C stands for constant region, okay. It is not going to change from cell to cell or from antigen receptor to antigen receptor. It is a fixed form, okay, a fixed genetic sequence, a fixed structure, okay? And we also have V regions. These are called V because they are variable regions. These are going to be distinct from cell to cell to cell to cell. They're going to be different. This is where the binding is going to the antigen will take place. And it's also where the specificity and the variability of each cell come from. Okay, so we have two different chains here. We have a light chain and a heavy chain. They're so named because their molecular weights are different, right? The more protein you have, the heavier you are. Okay, so the light chain is the shorter chain, and they're bonded via disulfide bridges to the heavy chain, and then the heavy chains are bonded here in the constant regions to each other to create this Y-like shape. Okay, so we have two antigen binding sites per B cell antigen receptor. Okay. Now, let's see. So when an antigen receptor of a B cell, this is sort of a broad overview, comes across a pathogen, okay, the pathogen can have many sites on it that are antigens, okay, and the specific site that it binds on the antigen is called the epitope. Okay, so a B cell here, there may be many epitopes on the surface of the pathogen, but this B cell will specifically bind only one of them. Okay. If it binds and gets activated through a process of binding and communication with some of the other helper T cells, then it can differentiate into what's known as a plasma cell, which will make clonal copies of itself that have the same sort of genetic material that express the same shape of the antigen binding domain here. And it will replicate itself, and then it will turn into a plasma cell, which can sort of replace the constant region of that transmembrane domain, that the platform that holds the antigen binding domain, okay? And it can basically cut that off of that constant region off and now create these soluble antigen binding domains known as immunoglobulins, which then also known as antibodies, which then get secreted out into the lymph or out into the blood. It will travel around and now bind to that epitope wherever it's found on any of these antigens. Okay, now these antibodies are highly specific to a particular epitope. Okay, they bind with extraordinarily high affinity usually measured in the picomolars. Okay, so once they bind, the binding is essentially irreversible. Once it binds, it will take days oftentimes for it to fall off. Okay, so this is irreversible binding to a particular antigen. Okay. Now, so this is basically sort of the overall process of B cell binding. T cells work a very similar way. Okay, they don't have this Y shape though. They just have a single stick that's sticking up, but they still have they still have two variable regions, and they have a constant region here which is formed by a which is sort of merged by a disulfide bridge. They have a transmembrane domain that displays it, and these two different chains are roughly the same length. Okay, they're called the alpha chain and beta chain instead of heavy chain, light chain. Okay, so they both T cells and B cells, their antigen binding sites are highly variable, but the constant regions are a little bit different, and they have a sort of a different function. Okay, this constant region is really important for a T cell because a T cell doesn't like to bind to an epitope by itself. A T cell has to also have that binding interaction stabilized by an interaction with a platform that the epitope is being displayed on. Okay, so what am I talking about? Okay, so within all of our cells that have nuclei. So which cells don't have nuclei? Red blood cells. Pretty much anything besides a red blood cell, okay, is going to have a type of protein called MHC, major histocompatibility complex, which are molecules that are going to display fragments of the proteins that are made and digested inside a cell onto the surface of the cell. This is like, telling your basic, because it's very difficult. An immune cell that's trying to find out, are you sick or not? Okay, would love to know what's going on inside the cell itself that it's talking to. But it's very difficult to know that, right? So instead, our bodies and our sort of immune systems have evolved this MHC molecule that will, whatever is being degraded within our cell, little pieces of it get displayed on the surface of our cells. Okay? And that happens in all cells within our body, except for the red blood cells. Okay. And this is called MHC class 1s, okay, these platforms. Okay, so when a T cell comes in, okay, it has to, okay, now there's a second class of MHC molecules that are going to be displayed only by a subset of cells in your body. These are called these are basically the professional antigen presenting cells for APC. Okay, so these are cells which will phagocytose things, okay, dissolve them and break them down in that lysosome, okay, and then display those fragments on the surface of the cell. But that is displayed on a different class of MHC. Okay, that's on a class 2 MHC. Those class 2 MHCs are only present in these phagocytic cells. So those are saying, not what am I making inside my body? They're saying, what did I just have for lunch, right? What did I just digest, okay? And the T cells can come in, depending on the class of T cell and depending on the class of MHC that it's being displayed on, can potentially bind to an epitope there and have that interaction stabilized, and that stabilization can cause the T cell to get activated. We're going to go into this in a little bit more detail in a second. So how do we make these sort of pathogen, how do we actually make these little fragments? We can do that through something called the proteasome. Okay, so this is basically, everybody's probably seen the garbage disposal in the sink, operates via the similar principles. Basically, the proteasome is going to take proteins within your body, and they can be either within your cytosol or they could be within these lysosomes, okay? And they will grab onto a protein and they will basically pull it apart and digest it into pieces, right? So here is a sort of protein. The proteasome will pull on the protein, sort of uncoil its amino acid shape, and then physically chop it into pieces of fragments, not individually, usually longer than a single amino acid, but chop into peptides that are long enough so that they can sort of retain some of the structural information that's embedded within that amino acid sequence. So they're still useful for identifying an epitope, but are no longer functional or pathogenic. Okay. Okay, so there's four major sort of characteristics of our adaptive immune response. So we're going to have huge diversity in the lymphocytes and those antigen binding domains of the receptors. Okay, but so that's great. We have to be able to recognize any possible pathogen. Okay, and we do. Our bodies can recognize any possible pathogen because of that insane diversity. But at the same time, we have to be tolerant to any potential epitope within that's produced by our own body's tissues, right? So how can we recognize self-produced versus externally produced proteins or molecules? Okay. Then we want to know that we want to show that the B T cells can amplify themselves specifically after they're activated, and then they can retain that memory of what of which cells proliferated. Okay, so let's look to see how this diversity of those binding regions is actually accomplished. Okay, it's a little bit of a complicated slide, so let's just sort of walk through it slowly over the course of a few minutes here. Okay, so the structure of any gene in a eukaryote, like humans, animals, okay, is going to consist of sequences of introns and exons. Okay, exons are areas that can turn into the protein, that can get translated into the protein. Introns are areas, the spaces in between those exons. Okay, they still have genetic material, but they never get translated into proteins. Okay, they sort of work as sort of structural regulators, okay, of how that DNA is going to fold and how it is going to interact with the processing molecules and the processing proteins of inside your cells. Now, an important step in the translation of DNA into protein is going to be the removal of introns from the sequence, okay, or from the messenger RNA. But in addition to that, specifically in B and T cells, there's an additional type of recombination that takes place that is going to allow a great diversity of underlying, a great diversity within the variable regions. Okay, so here's an example of the DNA in an undifferentiated B cell. So this is a B cell that's just been born. Okay, it's in the bone marrow. It has not matured yet. Okay. A B cell has what are called 40 V regions. These are individual exons that are spaced with introns between each one of them. Okay. Then it has 5 J regions. So the V stands for variable, the J stands for joining. Okay. And what will happen during the process of maturation of this B cell, there will be an event which causes recombination of the DNA between one V region and one J region. Okay, so what is recombination? That is where some machinery of your DNA processing, okay, is going to come in here and say, I'm going to cut out, I'm going to find a sequence that's very similar between here and here, okay, and I'm going to cut out everything in between. I'm going to paste these two spots together. Okay, it's like you're just taking out a loop. So everything between, in this example, everything between V39 and J5 gets cut out. Okay. And now we've pasted together V39 and J5 into a single exon. Okay. Now that single exon still has one intron before it gets into the constant region, okay, which is part of your heavier light chain. Okay, so this is, I believe, the light chain that we're looking at here, okay, because there's only 40 and 5, rather than a more, like the heavy chain actually has a whole nother sort of set of combinations that you can put here. okay? So we have, basically, we have this recombination event that occurs, and now this is now a differentiated B cell, okay, where this recombination event has occurred. So now we can only make, instead of having the possibility of making 40 times 5 different variable regions on this light chain, now it's only allowed to make one that has 39 and J5 stuck together, okay? So now this can get transcribed into mRNA. Okay. Then through the mRNA processing, we're going to cut out the intron, add these different sort of parts to it that'll allow translation of that mRNA into protein. And then at the end of the day, we're going to get a light chain polypeptide that's produced, which is the variable region, which is actually B plus J, okay, and the constant. So every B cell, they all use the same C. They all use the same constant region, but this V region is going to be a different combination of the V and J's. Okay. Yeah. The differentiation is thought to be random, totally random. So, and you know, why do we need to do this? Like, we are going to make, we have to have over 1 million different B cells, different variable regions on a B cell in order to capture all the potential antigenic diversity in pathogens, okay? But our entire genome only has like 20,000 cells or 20,000 genes within it. So we can't create one gene for each possible antigen, right? Instead, we have to combine genes in a way, in a combinatorial way that can dramatically expand the variability. So, you know, here we have 40 times 5. Those are 40 times 5 different possibilities. So that gives us 200 different sort of V regions that could exist on a B cell variable region, okay? If we look at, if we look at the heavy chain, they actually have a whole nother sort of set that gets combined here. They have like another region here, V, they have a D region for diversity, okay? And they'll combine a V, a D, and a J. We're just not showing it because it's kind of complicated, but it ends up getting even like thousands and thousands of different possibilities for your heavy chain. Now, if you then combine one light chain with 200 possibilities and then thousands of possibilities in your heavy chain, now all of a sudden you're getting up to hundreds of thousands or millions of different possibilities for the combined heavy and light chain variable region, which merge together here to form that antigen binding domain. Okay, so the total number of combinations that you can get between the heavy and light chain variable regions is 3.5 million. Okay. Based on just based on this recombination. Yeah. So do they randomly do this until one of them works? Yes. So each B cell randomly does this. Okay. So you get one of 3.5 million different possibilities out there. Okay. Just from recombination alone. Okay. And each one of them has, so now each one of them has its own signature binding pocket, right? So you have a huge diversity of this within your B cell population. And then only a few B cells will be able to bind a particular antigen and get activated by it, right? So then we have to find which ones of those bind that particular pathogen and then amplify those. We have to expand that line of B cells. So like, you know, we've got one of each of these, okay? And then we've got one that binds appropriately to the pathogen. Now we're going to make millions of copies of that particular one so that we can mount our adaptive immune response against that specific pathogen. Okay. Cool. Okay. So one other thing I must mention is that even once that B cell is recognized and starts to undergo the expansion, there'll be a second process called somatic hypermutation, which will then start mutating the sequence of that variable region, okay, in the copies of itself to create even greater diversity that's targeted to the pathogen itself. Okay. Yeah.
What is the light chain?
So the light chain and the heavy chain, the light chain is, so each antigen receptor consists of a pair of heavy chains and a pair of light chains. Okay, these are sort of fused together by disulfide bridges when they're expressed. Okay, so this variable region consists of one heavy chain and one light chain that are stuck together, and this forms a functional binding unit. And that is duplicated with the exact same specificity on the other side. Okay. It has two arms, each which has the same sequence. Yeah. Where does somatic hypermutation occur? We're going to talk about that in a second. It happens in germinal centers within lymph nodes when there's this process of affinity maturation that we'll look at in a second. Yeah.
Oh, you're gonna talk about somatic.
Yeah. Yeah. I believe so. Okay, I think that's in 3 slides. Okay. Now, so we've got this sort of diversity of B cells. Now we've got a problem. Okay. How do we make sure that the B cell don't attack our own, don't attack our own body's tissues? Okay, we want to mount an adaptive immune response only against exogenous pathogens, exogenous antigens. So when we're in the bone marrow, okay, we have our sort of immature or pre-B cell. It hasn't even turned into a B cell yet. Okay. Now it is going to sort of have that recombination event that occurs. It's still going to be considered immature, but now it's going to display the immunoglobulin, the Ig here, okay, of a random receptor, right? It's going to have its own unique signature now of its binding area. And now within the bone marrow, there will be a variety of internal of cells that get passed alongside it, okay? And if it binds to on those cells, it kills itself. It undergoes apoptosis. In the context of the bone marrow, if an immature B cell gets activated, that will trigger an intracellular response of apoptosis to kill itself because it is going to be tested for sensitivity to any of the body's tissues at that time. Only when it is shown that it does not bind to anything that is produced by the body itself is it allowed to mature and migrate out of the bone marrow into the lymphatic system and then be used to screen for pathogens. Does that make sense? Cool. Okay, so only a tiny fraction of antigen receptors are specific to epitopes on a given antigen. Okay, so why is our why is our adaptive immunity so effective? First of all, in the lymph nodes, an antigen is going to be presented to a steady stream of lymphocytes like B cells and T cells until you make a match, until you find that 1 in a million that really effectively binds that pathogen. Okay, then the second part of the answer depends on what happens when a B cell or T cell binds to that antigen. There's going to be a proliferation, a selection and a proliferation of that cell type due to the activation of that. That binding is going to sort of facilitate an activation and a clonal expansion of that B cell or T cell, make multiple copies of itself that all have identical affinity, just that specific antigen, right? So we've got millions of B cells that all are different our antigen specificity. The one that binds to a pathogen, okay, an antigen on a pathogen, that's going to get activated, make copies of itself, and the copies of itself will sort of turn into a reserve pool of B cells called memory cells, or it will turn into plasma cells, which are the little antibody factories, which will then churn out more and more antibodies that will attack and neutralize the pathogen. Okay, now let's talk briefly about affinity maturation. So when you first, you know, we have 3.5 million different possibilities for our B cells here. Okay, that's a lot of diversity. Okay, but we can do even better than that. Okay, that's just good to sort of pick out a few that can hit the pathogens strong enough to get activated. But then following that activation, okay, within these sort of masses of cells called germinal centers within the lymph node, once that activation takes place, we're going to want to try to create immunoglobulins or antibodies that are ever more perfectly fit to the antigen. Okay. Because 3.5 million is not enough diversity, actually. So now we want to take one that works pretty well and find ones that bind even better. Okay. So this is the process of affinity training, which works via somatic hypermutation. So B cell is going to get, this B cell gets clonally expanded, makes copies of itself. Okay. As it's making copies of itself, the section of the genome that encodes the antigen receptor mutates at a frequency of like 1 million times more than you would normally mutate. Okay. This is a really interesting process. It's still not fully understood how somatic hypermutation works, but it's absolutely essential for an effective immune response. So now, each one of these, so it's not changing the combination of V, EJ regions. Okay, you're set with those, right? You've got that one, you're set from the beginning, from your sort of during that maturation process, you're set with those. But now what we're going to do is we're going to change individual nucleotides within those regions, about 1 difference for each cell division. Okay. So that's just going to tweak the shape of that variable region just a little bit. Most of those mutations are going to change that binding pocket in a way that's not that useful or doesn't do much. Okay. But a few will change the binding pocket so it binds even better, it fits the epitope even better. Okay. Now, if you bind, and now the within this area, okay, within the sort of environment of this germinal center, okay, if you bind with lower affinity to those antigens, you are going to, those B cells will commit apoptosis and allow their sort of, apoptosis is really just a way of, putting yourself in the recycling bin so you can get, your material can get made to B cells. Okay. The ones with higher affinity, okay, will get activated by this coordinated response from a dendritic cell, an activated T cell, and then that's going to drive a differentiation of the B cell into either plasma cells or memory cells. Okay. So if you have the best affinity, you are the winner. You've got the highest affinity one, you are going to make antibodies. You are sort of destined to a new outcome where you're going to be the factory that's making the warheads against whatever that pathogen is. Okay. These plasma cells are going to start spitting out the antibodies that produce, okay, that go out all through your lymphatic system, through your bloodstream, try to find pathogens all over the body to kill them. If you have moderate affinity, okay, then you get turned into a memory B cell where you're going to be put into storage and try to recognize that specific pathogen, or maybe some slight variation of that pathogen again, when if after you kill the pathogen this time, it comes back in a few years. Okay, those memory B cells will sort of lay in waiting to provide a very rapid response against it. So what does a plasma cell look like? Basically, this is the shape of a plasma cell. Okay, or this is a electron micrograph of a plasma cell. What we can see here is that you have a huge amount of endoplasmic reticulum. It's basically all nucleus with sort of a chromosome, chromatin here, an endoplasmic reticulum, a few mitochondria just to provide ATP, and it is just constantly spitting out immunoglobulins, these soluble immunoglobulins called antibodies. They will produce about 2,000 antibodies per second over their lifespan, which is about 4 or 5 days. So billions, okay? So that is the role of a plasma cell. So you want the highest affinity ones to be making those antibodies, okay? Now, if we look to see how we can also measure the antibodies within the blood, okay, to get a sense of how far along in an infectious process are you and how strong is the immune response that you have to that particular pathogen. Okay, so then this is sort of a process of immunological memory. And So if you are exposed to antigen A, okay, how long does it take to start making antibodies for which you can actually see that in your blood? Now this is a log scale here. Okay, so this is 10 times more than this is 1, this is 10, this is 100, this is 1000x. Okay, so it takes 3 to 5 days before you really can start turning on, where you can get through this process of affinity maturation and start producing plasma cells that are going to be pumping out soluble antibodies. Okay. And then those antibodies are going to go and attack the pathogens. We'll talk about exactly how they do it in the next lecture. And you're going to have a peak response here. Probably you kill the pathogen. You sort of clear the infection after 2 or 3 weeks. And then if you're exposed again to the same pathogen that has antigen A, you have this huge pool of B cells already there that have, instead of having just 1 or 2 cells that can recognize the pathogen or antigen A here, you've got millions just sitting around in your B cells. So they can mount a much faster response, only take a day or 2, and a much more vigorous response, 1,000 times stronger response, which is why it's so effective. But this is specific to the antigen. This response, if you expose yourself to a different antigen B on this other type of pathogen, you don't get a bonus for that, right? Because you have not yet trained your body or immune system to produce antibodies for antigen B. Okay, yeah.
For like the second exposure to the same antigen, well, antigen, yeah. Like, is it a memory cell that able to like quick response, or is it?
The memory cells can rapidly differentiate into new plasma B cells and then release that. But you're starting, instead of starting from 1 and clonally expanding it, you're starting from thousands of cells. So you're starting from a much higher baseline. Other questions about this? Okay, so this is the yeah. That's a great question. So how long does this last? How long does this memory last? It really depends on the class of pathogen and the antigen type. And there is a fading of immunological memory over time. But if you space out the presentation to that antigen a few months or a few years apart, you can build up stronger and stronger responses to that antigen over time, right? This is why you have things like a vaccine schedule, where a vaccine oftentimes is a degraded or destroyed sort of form of a virus or pathogen that trains your, mixed with an adjuvant, which is just a molecule, sometimes it's aluminum, that's going to sort of get your immune system kind of pissed off, okay? And like, oh, look, this thing just got injected into me. Okay, I'm going to try to mount an immune response against it, okay? So I can train myself to make antibodies against that antigen. And then if I inject myself with that same vaccine 2 or 3 years later, you get an even bigger response. But over time, those responses do fade. Okay, so there is, and that duration of fading could be usually measured in years. Okay, and they can also be impacted by other diseases which can interfere with your immunological memory. So things like measles are known to actually cause a reduction in your ability to mount immune responses across to other antigens. Okay, so that's all for today. We're going to go into greater detail on MHC proteins and how these tools actually function against pathogens on Thursday. Oh shit.
I just have a quick question. Have you
Yeah. 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 Let me just shoot anytime, really, but you can always send me